In our last unit, we used an MR session as an example of a common and complex type of data that XNAT supports. This wasn't by accident. One of XNAT's first mission statements was to provide a better way of managing image session data for research purposes. The alternatives that were out there, such as a PAX system or storing things on a local file store, were just simply insufficient. What makes image session data so challenging? Several things. For one thing, the data itself is huge, particularly when compared with genomic data or other types of statistical data that are typically gathered for research purposes. This not only impacts your file storage capacity, it also requires significantly more computing power to run processing algorithms on image data compared to these other types of data. For another thing, the data is complex. You have a blend of 2D pixel data that's stacked in three-dimensional stacks that all get combined to form a single scan. And each one of those files has indexed data stored in DICOM headers. And all of that has to be read and associated with each other in order to complete the concept of an image session. Also, the data can be messy. You've got scan artifacts that can come from subject movement or a misaligned setting on the scanner or any other factor. Image resolutions can vary wildly between modalities, which makes comparisons difficult when they're possible at all. And image acquisition has to be accompanied by a quality check to make sure that the data is usable. And finally, if you're not careful, storing the data itself can get you in trouble with your institutional review board. Clinical imaging systems were designed to capture patient data, and in any kind of human subjects research, that kind of PHI is verboten. In this unit, we'll discuss each one of those challenges, and we'll talk about how XNAT was engineered to help you handle them and to support your imaging research workflows.